kind of distracted in presenting, but unfortunately, we have to bear with it.
I'll be ready. There's Dennis.
Yeah, your friend Dennis joined. I sent it to him today morning saying, hey, your buddy Tom is here.
I'm looking forward to the next one too. It sounds like an interesting topic.
Yeah,
Yeah.
we'll continue to have, because nowadays we are getting a lot more people from different areas coming in, and it's like more knowledge sharing, and how this AI is. It's interesting, how these area, even we got from SenzMate point of view, we got into this domain 5-6 years ago or more, but now it's becoming with everything. This is I call it low hanging fruit because of the data and all that. So it's interesting.
It is.
And you've been doing so long on that. It's different and
Different domain.
Yeah, it's interesting to see in the real life use cases. So.
Yeah. So actually, we have some good number of people already joined, so probably we can kick off and start.
Yeah.
Right. One. So thank you everyone for joining on time. So there are people joining. So hello everyone. Welcome to our webinar number 6. This is in our series. So this time it's a very fascinating topic to talk about. So we are talking about impact of AI in life science. I am Miller Rajendran, and joining with my co-host and moderator, a veteran entrepreneur, Ganapathy Pillai. Today, we'll be exploring how artificial intelligence is revolutionizing the life science with the focus of genomics and drug discovery, diagnosis, and personalization of medication. These advancements are shaping the future of healthcare and driving innovations and improving patient outcomes. So before getting into everything and our esteemed speaker, quick note for the audience, this session being recorded and we'll be sharing the link afterwards. And please feel free to send your questions and your thoughts in Q&A or in the chat box. And feel free to raise your hand. All the questions will be answered in the tail end of the session. So thank you so much for joining again, and over to you, Ruben.
Yeah. Thanks, Miller. We are thrilled to have my good friend Tom Schaffer as our speaker today. Tom has an impressive background in high-performance computing. First, I met Tom at an Edge AI conference in Silicon Valley a few months ago, where he presented some of the content we are going to discuss today. Since then, I've been interacting with him and learning about how the life sciences evolved over the last 20-30 years on using these kind of AI and big data to have impact on drug discovery, pharmaceutical areas. So it has been a fascinating discussion with Tom. So that's why I invited him for this webinar. And to give some background on Tom earned his PhD from Carnegie Mellon University in computer engineering with focus on parallel computing architecture and programming models. His interest in high-performance computing took him to NASA Ames, most prominent research area. And then he went to Rambus, where he worked on accelerated memory interfaces for providing high bandwidth. If you look at the big data and amount of data, you need to have this kind of the data fed into the system. But that's where you need this high bandwidth. Following that, he co-founded the cloud video analytics company Sensor.net that applied scalable cloud computing to analyzing large streams of video data. Again, that's another groundbreaking area. Then he later joined Roche Pharmaceutical to work on next generation sequencing and scalable genomics analysis platform. Throughout his career, Tom has focused on application of high-performance computing system to real-world problems. And with now all these AI, GPU, and computing, and those all collide together, come together. So with this, I will let Tom to do a quick intro of yourself. I gave you some of them, but you can give you of your work experience part of it, and then go into, how we are going to do today is Tom is going to go through about 20 minutes presentation of the content, and then we will have some Q&A, and then we will, as Miller said, we would like to get your audience questions, and Tom will be there to answer that also. Over to you, Tom.
Yeah, Ruben, thanks for the really nice introduction. I'm thrilled to be here today. I think it's going to be an interesting discussion. I think you covered most of my background. I'm interested in how high performance computing can be applied to real world problems to benefit people. And that took me to sequencing and healthcare, where advanced computing is having a direct impact on people's lives. And that's kind of exciting. So let me start my presentation and make sure it's the right size. All right, is that showing up? All right, so the title of my, we set?
No, all good. You can take it forward.
Yeah, so today I'm going to talk about AI and ML challenges in genomics. And all right, so the preface, my goal is to give insights into the characteristics of genomics computations and explain how AI and ML on the edge is used for DNA processing. What I'm going to do, I'm going to talk about challenges in AI and ML for genomics from the real world. So I got involved in genomics a few years ago, maybe 2017, and shortly after that, I was motivated to apply computing to problem areas that can impact people's lives. And there was this article in the Washington Post about whole genome sequencing being applied for newborns. And there was this really dramatic case of a young boy, he was 6 years old, having seizures that were difficult to diagnose. But by applying whole genome sequencing with a process that took about 39 hours to perform, they identified a simple treatment. And this was a few years ago, but this really resonated with me. And what's interesting is that computing technology is a big part of dropping this turnaround time to 39 hours. Now, the bread and butter of sequencing is not for cases that are as dramatic as this. Most uses are for cancer screening and diagnosis, which are also very important, but don't have the dramatic turnaround time requirements that this young boy did. However, for other people, waiting for a diagnosis, having rapid turnaround is really important. And so what we're seeing is the application of advanced computer architectures and improvements in computers is reducing the turnaround time and helping people faster. All right, so some trends. In 2006, it cost about $300,000 to perform whole genome sequencing. That got down to about $1,000 in 2020. And finally, this year in January, Ultima Genomics released a sequencer called the UG100, and they claimed that they had the $100 genome. This is making whole genome sequencing available to many more people for more types of cases. And so what we're seeing is that there's a decrease in cost that's faster than Moore's law. All right. So in the past few years, we've also seen a new generation of sequencers released from a number of manufacturers. Ion Torrent, Illumina, PacBio, Element, Ultima, Singular. And I want to point out the middle column. The middle column here is identifying the output per run, the amount of data that a sequencer produces. And the metric is gigabases. A base is one of the letters of DNA, A, C, or T, or G, but it takes about a byte to represent a base. So you can read this as gigabytes of output. And what you'll see is that all of these sequencers are producing output folders in the order of hundreds to terabytes of data. Now, all of this data needs to be transferred, analyzed, sometimes compressed and stored. And this capability, with the addition of greater volumes of data, more computing power is needed to keep up with this data. Now, to contrast, a previous generation of sequencers, maybe 5 years ago, were producing about 10 to 15 gigabytes of data. So this is a dramatic increase, but what's made it possible is advances in computing technology and also GPUs. So note about the sequencing market. Currently, the sequencing market is about $18 billion. It's expected to grow to about $60 billion in 2030. About half of that is consumables. Consumables are the reagents and chemicals used for performing sequencing, what I call the wet stuff. But there's an increasing portion of this devoted to the platform and software. Those are the top two bars. And it's becoming significant. In the construction of a sequencer, what I've observed is that there's more high performance computing capability, multi-core processors, and a number of GPUs driving up the cost of sequencers. But this is also an opportunity for people like me and us who work in computing and want to apply it to this problem area. There's a need for engineers who know about high performance computing, GPUs, neural nets, and machine learning. And it's increasing. All right, so what I want to do in this talk is give an introduction to sequencing. And the reason I want to do this, I want to explain for people who know about software but don't know much about sequencing, the characteristics of some of the algorithms and operations that are performed. And that'll help explain how AI and ML are being applied. So a sequencing workflow begins in the lab with extraction from plasma. There's a step called library prep. Samples are pooled. They're finally inserted into a sequencer, and this is where the really interesting step of turning real-world samples into data, the virtual world, is performed. And then finally, we'll talk about the analysis pipeline. These are all the steps of computing, and I'll talk about how machine learning algorithms and AI are being applied in there. All right, so the first step is extraction. Extraction treats a sample of plasma and extracts its strands of DNA. And just to review, DNA is a double helix, and the elements of the double helix are nucleotides, and they're denoted by the characters A, C, and T, and G. So what we're doing in sequencing, we're looking at the DNA strands of a patient, and we're looking for anomalies or mutations. And these anomalies correspond to diseases or conditions that we want to diagnose or treat. So a lot of these operations will end up looking like string operations on sequences of letters. All right, so the very first step to prepare the DNA strands is to fragment them. So we start with a strand of DNA that's called a template, and that's broken into fragments chemically. Now, library preparation attaches adapters to each of these fragments, and an adapter has a coding part that identifies a patient. So for example, if these were fragments of my DNA, we might attach a fragment ACAC that corresponds to Tom's DNA. And then there's a yellow portion, what I've shown here in yellow, that's a coding portion that helps the sequencer identify the beginning and end of the fragments. So we've taken DNA fragments and we have attached little sequences, synthetically generated DNA. We attach them to the front and the end. And this does two things. It helps the sequencer identify the start and the end, and it also identifies which patient these fragments are from. All right, so then the next step is pooling. In pooling, we combine the fragments, from different patients into a pool. Now, a pool, it's a physical container. It's like a test tube, and we're actually taking samples and pipetting them into a vial. We're using a machine to do it. But the reason we do this is because sequencing is very expensive. And this is a form of time sharing for the sequencer. We're going to have numerous patients' DNA sequenced at one time to save on the cost of reagents and to amortize the cost of running the sequencer over these many patients. All right, and then finally we get to sequencing. So the sequencer takes as input a pool of fragments with the labels, identifies those fragments, reads the letters of DNA, A, C, and T, and G, and produces data files. Sequencer run takes between 12 and 48 hours. And as I explained earlier, the amount of data produced is somewhere between hundreds of gigabytes to now terabytes of data. So this is an amount of data that's large enough that it stresses our current computing. And of course, we can always use more bandwidth and more capacity. All right. So I'm going to switch gears and talk about the data pipeline from the beginning inside the sequencer with the sensor through to the end. The first step is called primary analysis. This is where we read the letters of DNA in the fragments. The next step is demultiplexing, where we separate fragments from different patients into separate files. Consensus is where we take fragments and reduce the number of fragments, but increase their quality, and I'll explain that later. And then finally, variant calling. Variant calling is where we look at the fragments, identify where there are anomalies against an expected human expected fragment from healthy humans, and identify things that could be a mutation or a variant. All right, so one primary analysis. Primary analysis is where the real world is sensed and it's turned into data. So one type of sequencing is called nanopore sequencing. And in this, a nanopore is a minute molecular structure that allows one strand of DNA to pass through at a time. Now, by applying an electrical voltage over the nanopore, as a strand of DNA is dragged through the nanopore, we can sense changes in current. And so this picture illustrates a strand of DNA passing through a nanopore with the nucleotide corresponding to a C. What the sequencer would see would be a change in the current over that nanopore, and it would have a level corresponding to a C. Now, one way of identifying what the nucleotides is by using DSP algorithms, but increasingly we're seeing neural nets and machine learning applied to this task. All right, so what's the challenge with using machine learning for base calling? Well, one is keeping up with real-time constraints. The chemical reactions that are producing data, whether it's nanopore or other types of sensing, occur in real time, and having GPUs keep up with that can be a challenge. For machine learning, runs are expensive, and if the scientists are changing the chemistry often, then the data is going to change. So in training machine learning, we need to have good data, and it takes time to train. So it's a challenge producing runs because of cost and keeping up with producing data sets for machine learning when we're developing a new model. And of course, in sequencing, the data is inherently noisy. And machine learning is good at working through the noise, but it's still a challenge because sequencing is inherently inexact. All right, the next step is demultiplexing. And in demultiplexing, we're taking a group of fragments from multiple patients and separating them into separate files by looking at those tags at the front and the end that I showed earlier. Another way to show this is that we have a set of reads that are coming out, and each of them has a prefix, and we want to separate them into separate files for the reads that have the same prefix. So in the past, people used string type of algorithms to perform this operation, but increasingly people are considering using machine learning. But there are challenges applying machine learning to this process, and that's because error rates from the previous step stack up. So look at the picture at the bottom. This is showing an adapter length 40, excuse me. Now, if the base calling accuracy of the previous step is perhaps 99%, that corresponds to Q20, and the adapter length is 40, then the probability that the entire barcode sequence is correct is only 68%. So this is a machine learning algorithm to do demultiplexing would need to work with a barcode of a accuracy of 68%. Now, it's more typical to have higher quality, but still the challenge remains. It's noisy data. It's difficult to deduce the actual prefix sequence in the face of all this noise. So Oxford Nanopore published an interesting paper where they said, look, what if we combine base calling and demultiplexing into a single step? So the top of this chart shows a more traditional algorithm where there's a raw signal coming in. The base caller is essentially quantizing that raw signal to levels corresponding to individual nucleotides, ACTG, but with noise. And then multiplexing is being applied. But Oxford Nanopore said, look, what if we apply these two steps all at once? maybe we can get better results. And they do. And what's interesting about this to me is that this is a new formulation of the problem enabled by machine learning. The more traditional pipelines couldn't be fused in this manner. And so what we're seeing is that the availability of GPUs and machine learning is enabling a new class of algorithm for this process. All right, the next step in the pipeline is called consensus. And in consensus, we take a large volume of reads and produce a smaller number of reads with higher quality. And the way we do this is by gathering reads that look like they're from the same place and then have them vote on what they think each position actually is. So if all of the fragments agree on the value of the letter, the nucleotide in the same position, we consider that truth. And if they disagree, if the disagreements are small, then we might chalk that up to a sequencing error. So one well-known algorithm for performing consensus is called deep consensus. I think this came out of Google, and it's a machine learning algorithm. What's interesting is a few years ago when this was released, and PacBio ran an analysis, they could perform consensus on the output of one run in 500 hours. Now, what was interesting was that there was easy parallelism, and so they could do it in 1 hour with a total speed up of about 3.3%, 3.3x. However, PacBio has a new machine called the Revio, and what they're doing, it seems, is that they're passing through high quality reads as is, but for lower quality reads, they're applying consensus. And so that in an average run, the total amount of processing power devoted to consensus on just the poor quality reads adds 5.5 hours to the run. And there's this interesting result here on the right. Even though performing consensus is taking 5.5 hours on the low quality reads, they're reducing the runtime from 30 hours to 24. So the low quality reads that would have been thrown away in the past on this new architecture are being improved through machine learning with the consensus algorithm, and the total time is still going down. So this is a pretty dramatic result. It's also interesting to think about this hybrid algorithm with high quality reads passing through as is, and low quality reads being improved through machine learning. Okay, and the last step in a typical workflow is variant calling. And calling here means identifying. So we're identifying a variant, and a variant is a place in a DNA read that looks different from what we expect in the expected human reference genome. All right, so this is what variant calling looks like. We have a human reference genome on the top right here, and we have a collection of reads that we got out of the sequencer. We have demultiplexed, and we've performed consensus on to improve the quality of some of the reads that weren't so good. And now we try to map the reads to where they should go, where we think they're from in a human reference genome. And with each mapped read, then we look for positions where it differs from the expected human reference. And these correspond to variants or mutations. And some of them may correspond to a disease or a condition, and that's where a genetic counselor may come in to interpret what these variants mean. So that's the operation. Variant calling is also being accelerated with machine learning, and GPUs and machine learning are enabling new classes of algorithms. One well-known family is called NVIDIA Parabricks. Parabricks was a company, I believe, it was acquired by NVIDIA that has accelerated many of the steps of a typical genomics pipeline. And they claim to have up to a 65x acceleration with the germline variant caller on GPUs. This has reduced the variant calling step from 36 hours to 33 minutes, which is pretty remarkable. But as an aside, because of the expense of GPUs these days, the total cost may go up because a minute of GPU time is so much more expensive than a minute of CPU time. But that's well-known, some very widely used algorithms called Parabricks that run on standard NVIDIA GPUs. There's another way that variant calling is being accelerated. Illumina has a system called Dragon. Dragon, I think, had its genesis in a company called Edico that Illumina acquired, and they were accelerating genomics pipelines using FPGAs. And what's interesting is that Illumina has enabled Dragon ML variant calling on runs as an option. And when enabled, it adds about 10% of the runtime. But by exploiting more available read and context information, it's leading to better quality results with more information. Another interesting use of GPUs to produce better, higher quality results. All right, so I'm going to talk a little bit about some trends of deploying these algorithms. So a few years ago, it was typical to perform most of this analysis in a cloud or perhaps a data center at a larger company. And so it's typical to think of that primary analysis, the reading of raw signal, converting into base calls, was being applied on the instrument. But all the further steps, demultiplexing, alignment, consensus, and variant calling, would be done in the cloud. So what we're seeing with the advent of GPUs is that more of these steps are moving onto the instrument or very close to it. So primary analysis, demux, alignment, consensus, variant calling, all of these have been accelerated and moved onto GPUs that are near to the sequencer, sometimes right on the sequencer. Now, what's been moved to the cloud is interpretation and therapy. Interpretation is where someone looks at or an algorithm looks at the variants and maps them to a diagnosis or treatment. Machine learning is also being applied in that step, but I won't have a lot to say about that right now. So to understand some of the challenges in using GPUs in a lab, it helps to look at what a lab looks like. So in a laboratory, we have operators. They're working with instruments. They're doing operations on pipettes and test tubes. I call it all the wet stuff. And they're in a temperature-controlled clean room, and operators may spend a long time working with the machines. And it's definitely not a server room. So if you're a software developer and you're thinking of deploying algorithms that might run in the cloud or in the server room, this is a different environment. They're going to run right there in the lab next to the operator. So this introduces some new challenges. So along the lines of heat and noise. So GPUs generate a lot of heat, and sometimes more than some labs can cool. And also, some of them are requiring more power than labs are typically set up to have. And also, the noise of the fans cooling the GPUs can exceed sound allowances for operators. So these are new problems that labs haven't been set up for, that the introduction of advanced computing and GPUs near the sequencer have introduced. So one solution is to split the sequencer into two parts, a wet part and an acceleration part. And I believe Ultima Genomics does this. They have a sequencer, they have an acceleration unit, and I believe that it's possible to deploy them with an optical cable connecting the sequencer to the acceleration unit. But this introduces another type of headache. IT departments may not be ready to route optical cable. They've probably set up their lab with Ethernet and switches, but now this is a new type of requirement, a very high bandwidth connection to the acceleration unit. So there are new challenges being introduced to sequencing labs by these technologies. Another challenge is deploying software updates. And there are two types of challenges here. One is updates in a clinical setting. So in a clinical setting, operators want to validate the software updates and validate that the sequencing operations they're doing are producing the results they expect, all the way from the chemistry down to the data. And so operators may tolerate updates on the order of 6 months. And this may be very different from the way a software developer or a machine learning engineer is thinking, where training new algorithms and deploying often may be the norm. In this case, in a clinical setting, that may not be acceptable. Another challenge in deploying updates is connectivity and size. The bandwidth to labs is continually improving, but it's not what you may be used to in other industries. So deploying model updates and software updates can be a challenge, just getting it into the lab. And also another very big challenge is obtaining data sets for training machine learning models. There are problems with privacy. It's typical for scientists performing machine learning training to need to work through the privacy of the data they're obtaining. And so it's typical to need to develop a way to anonymize the data, because genomic data is very, very personal data. In other cases, the data may not be anonymized, but will obtain a patient's consent. And consent, I think, as we're moving forward, it used to be the case that we just obtain a patient's consent to, quote, analyze the data, but now a patient may want more control over that. What are you analyzing it for? How long do you need access? And can I revoke access? And these issues of privacy and consent haven't been resolved. Another issue is population bias. For many of the data sets that are available, they may represent a particular population from a particular area of the world. And so we need data sets from other populations in other parts of the world. And as I said earlier, there's inherently noise in data. And so these data sets will have noise, and that's just part of the problem. All right, so in summary, genomics computations are inherently parallel. And this is really good news if you're a computer engineer. There's parallelism at the read level, there's parallelism at the patient level. So GPUs are able to exploit this parallelism directly. I also think that there's an opportunity to introduce streaming architectures. So the way many pipelines are set up today, they work in chunks. They get a chunk of data off the sequencer, they perform the first step in a chunk. It's interesting to think about streaming the computations on each read as they're produced. And this would require essentially a big loop fusion transformation on the pipeline. And we're seeing some evidence already, but I think that there are interesting opportunities for advances in bandwidth and connectivity to enable a new type of architecture that's streaming. But as I said, there's challenges in thermal, noise, and power deploying current GPUs. There are challenges with deployments and updates, and there are challenges obtaining trade-in data. But overall, machine learning and GPUs have enabled stride in turnaround time and processing more data. And both of these have improved patient outcomes directly, which is pretty exciting. All right, that's all I had to say.
Thank you so much, Dr. Tom. And it's really fascinating presentation. And it was a real deep dive into the genomics and sequencing and the related computation advancements which happens in the recent past. And the challenges which is not many people touched on and address much. So it's really grateful to know all these contexts from the best person in town. So, Dr. Tom, I have a couple of questions. I'm a real fan of edge on this segment, and considering the pros and cons on the cloud versus edge, what are the factors that determine whether we go cloud or whether we use edge computing is preferred for the genomics application? Could you give couple of examples or some factors to this?
Yeah, so it's complex and complicated and related to budgets and infrastructure in some cases. So, I think maybe a decade ago when these computations were being moved to the cloud enabled great horizontal scaling and pretty, a lot of compute at a competitive price. And that's still the case, that you're able to scale out the computations. There are languages for expressing genomics pipelines that help programmers take advantage of that. But in some cases where there's limited connectivity to the cloud, these data sets, I said, are very huge. So moving from 10 to 15 gigabytes a few years ago up to terabytes. Transferring a terabyte out of a lab, or if you have multiple sequencers, transferring multiple terabytes of the lab up to the cloud to perform the biggest step can be, the earlier steps can be prohibitive. What I mean by that is that if you look at the pipeline, at each stage, the amount of data has been reduced, but the value of the data in some ways has increased. So there's raw read data, that's just raw signal. The next stage is to identify base calls, and that reduces the amount of data. The next stage is to demultiplex into separate things, and then consensus, and you're further reducing the data. Finally, when you get to variance, there's hundreds of megabytes or megabytes of data. So, so two things are happening. I think that while bandwidth and connectivity are improving, if labs can afford GPUs or even just one coprocessor, that's enabling performing some of the earlier steps right next to the machine. And that's what the manufacturers have done. They've moved the processing of the raw base calling and some of the consensus operations right into the sequencer, so that what's coming off of it is higher quality data that can be more easily managed. And then you can either send it to the cloud or send it locally. But the trade-offs are changing continually. It's always a factor of availability. I mean, currently it's a factor of the availability of GPUs and the bandwidth that a lab may have to the cloud. And yeah, it's pretty interesting to look at those trade-offs, and it's changed, right? So no one had the processing capability next to a sequencer a few years ago to do any of these steps, and it's been a rapid change, I'd say, in 5 years. That's really changed what's possible to do in the lab computing wise.
Thanks, Tom. I think it was very, very educational, as well as some of the reflection I was having when you were talking about all that in 1990s, when the cell phone market and the first level of wireless communication was coming out. I was working on some of these. Like, there are so much information comes out. As you said, there are a lot of noise versus there are good data. How do you separate them? And that's how the GSM wireless 1.0, all of them came out. I remember working on some of the Viterbi decoding and different segment, which was very interesting to see, see that part of it. And I see the amount of data, amount of things come out of some of these Thermo Fisher or some of these development tools. And how do you move that one, and what information to do at the edge versus what to push it to the cloud, and how do you manage the cost, how networking, isn't it so much of it there?
Yeah, maybe part of it is the maturity of the algorithms. When you're experimenting and developing algorithms, having a parallel computing in the cloud is the right way to go. But as you become more confident in the algorithm and want to commit it to hardware or GPUs, you can invest in that as long as it's not changing so much. And you were talking about cell phones. It made me think about the state of video maybe 15 years ago. And we had software codecs, right? And I remember transcoding something that would take hours. It was not real time, but we have codecs in everything now, right? There's H.264 codecs and there's AV1. I think Google's is called AV1. And they've been committed to hardware. So what happened is, as the algorithms have matured, it became reasonable to commit them to hardware. And I think we'll see, we're seeing some of that in genomics, and I think we'll see more of that as some of the operations become more mature and more understood and used in the same way by more researchers. We might see operations like compression. I didn't talk about compression, but storage of data is important, and we'll see that committed to hardware. And maybe we'll see some of these operations that I was talking about. Maybe consensus and variant calling committed to hardware. I mean, I think Illumina Dragon is already doing that in FPGAs, but we may see ASICs performing some of the key kernels of these.
No, absolutely, you are correct on that area, because when we look at SenzMate AIoT lab, that's how the formation was happened, because of sensors create so much data. That's where the
Right.
SenzMate part of it come. And how do you utilize that efficiently while you don't throw out the good data,
Right.
but don't accumulate so much data, it become cost prohibitive. Yeah.
Yeah.
So all of that. So to follow up on earlier example you gave about the patient and all that, in diagnostic, these AI tools are making early detection more accurate and available. How do you think that clinicians are responding to this? Because it is a kind of new thing to them, and one way they could look at it as a threat, other way they could look at it as because they are doctors and physician, and now saying, hey, somebody is telling me to do which is not what I was taught in my school days. How do you think clinicians are thinking about this?
Yeah, and I can only give my impression of those that I've talked to, but I think with they're looking at it with expectation but hesitancy too, because what happens in life sciences is that for any algorithm, there's a period of, whether it's clinical trials or validation of that would be of drugs or treatments, or validation of algorithms and analyses. It takes time, and I think that scientists need to see repeatable results. I also think that in sequencing, a lot of that happens in a very large open source community too. So there are open source data sets or open source algorithms, and the scientists are writing papers and comparing results. And so what it will take is a series of publications by researchers all around the world, committing to a similar or the same algorithm, perhaps the same algorithm running a model in a container on a GPU, a particular GPU operation, where a large number of scientists are saying, we agree on this technique, and the results that we're getting out of these reference data sets are what we expect. And so I think that that's, there's a technology component, but there's a research and science component that researchers need to start seeing these results in peer-reviewed journals for it to become acceptable in the healthcare setting.
Great, Tom. So I think we have to keep the time also in mind. So we have maybe another 2-3 minutes. But before that, I have a brief question. This is usually we have it in our webinar, the last 6 session itself. So there is any global or regional standards or guidelines in the interoperability of and the accuracy of AI-driven genomics analysis pipelines, and how these standards are influenced cross-border research collaborations? So these are especially on the compliances context. We always have this question when it comes to medical data.
Yeah, I'll be honest, I don't know about standards for validating the accuracy. What I can explain are some of the standards for making reproducible experiments on data. And in the genomics world, there are some languages that are standardized for expressing computations as a series of processes, essentially a graph of operations, with ways to have each of the steps of the operation be completely reproducible by encapsulating the algorithm in a container that's checked into a repository. What this has enabled is that a researcher in one lab can check out an entire pipeline, which consists of a series of container operations, and maybe they're running it on their cluster in their lab, and they can run it on their data set or public data set and evaluate the results. And another researcher in another part of the world can check out the exact same pipeline, the exact same containers, but maybe run it in AWS in a different sort of scientific computing configuration and have completely reproducible results there. And so in the genomics community over the last decade, there's been a lot of work in that space. You can look up the GATK reference pipelines expressed in these standard languages, and there's a whole body of work for enabling scientists to have reproducible science, data science across the world. And it's really inspiring what that community has done along that way. And I think that and it's already happening, that the algorithms are migrating to GPUs and machine learning, and people are sharing models. And the same thing is going to happen, that the machine learning based models will be more understood. And as researchers can check out the models in their own environments, they'll be able to validate the results.
Yeah, thanks, Tom. I think at this point we will start taking some questions from the attendees. It looks like there is Anju is asking some question. Can we unmute
Yes.
Anju?
Anju, you can, Dr. Anju, you can talk.
You can unmute. Yeah.
Okay,
Yeah.
thank you. Can you hear me then?
Yes.
Yes.
Hi, Andrew.
Okay. Hi there. Thank you so much for the presentation. I know in the beginning you talked a lot about how genomics is used in the field of cancer, and you provided a couple other use cases. I was wondering, is there research on using genomics in the field of mental health? Like, are you able to use genomics to identify like any mental health disorders, or just curious about that, if there's any overlap there?
That's a great question. And I'm sure someone is, but I'm not aware of that work. But it does make sense, right? That, as as variations in DNA can be linked to a particular condition, mental conditions are certainly could be part of that. But to be quite honest, I haven't read those papers or studied that. But I think I think that's a really valid.
Yeah, I mean, I think Tom, you're absolutely right, because looking at the pandemic or whatever happening with the mental health, and a lot of it has some hereditary part as well as environmental part. So I'm sure, but as you said, we don't know that as much.
Right.
But these are the areas. I mean, I always say that healthcare area is the low-hanging fruit for all of the new tools we are developing with AI and ML and all that. And I think you hit it at the right area there. And there could be, I think, where I think you are in East Coast, there may be Washington, D.C. area. People may be investing more money on some of these areas. And our hope is this can be easily addressed rather than ignored for the mental health, and help these, when the younger age to make it better. Yeah. So.
Yeah. So, and I do take that back. I am aware of work done to study autism. Just look for correlations with genomic markers on that. So I'm sure that there has been work to apply it to other.
All right, thank you so much. I'm a psychologist in the Washington, D.C. area.
Okay.
Okay.
I'm interested in mental health.
Yeah.
Yeah, and sometimes genomics is just, for many analyses, genomics is just one mode of analysis, and so it's really exciting to think about multimodality. So whether those are physical samples or periodic check-in with a patient about their condition, or maybe there's some way to monitor their behavior or their health. And what machine learning and AI is beginning to enable is correlation of those observations along with changes in chemistry or physiology. And I think we'll see more of that. We're already seeing that, but there's enabling those multimodality analyses.
Yeah, but to add to that, and one other thing, because I am in Silicon Valley, I see a lot of companies come through and different areas. There are companies who are addressing some of them. Maybe they haven't done deep research on the, cure part of it, but at least the symptoms to help with mental health, a different thing. There's a company called MeCare. There are a few other companies like that who are trying to be, build a companion, AI companion for these people, and talk to them early rather than wait when, they talk about crisis line, people make calls and all that. Those are already at the later stage. If we can do it earlier, detect some patterns. Like one of the things they were doing in that was they were listening to people's voice and as you speak, and it started moving the vibration changes and all that. They have some mental breakdown coming up, and they can detect that one, and advance it earlier to alert to the family members or the physician. So there are a lot of work is going on, but it's interesting.
Yeah.
Thanks.
Question,
I'm
thanks.
thinking it would be particularly impactful for like suicide prevention.
Yes.
So yeah, you mentioned like the crisis lines, that's like a late stage, but yeah, I was thinking especially for like veterans and service members. So I'm trying to, design some sort of solution, suicide prevention solution, but I guess I'll try to do my own research into it and see. But before today, I hadn't really thought about, honing in on the genomics aspect.
I've just talked about, analysis of DNA, but there have been companies that have used iPhones in different ways as data collection devices to monitor people's behavior or health, whether it's their eye health or in their mental health. And so, sometimes there's interesting things you can do with the sensors on an iPhone to observe how people are responding. Maybe there's a test you can do to measure their response time, and you can chart
regular check-in kind of thing.
Yeah, and have an AI correlate changes in those behaviors that may become predictors for the things you're interested in, Anju.
Yeah, so one other thing, Anju, is like we AIoT, SenzMate AIoT lab, where we do a lot of prototypes, testing, building early algorithms and all that. And if there is an opportunity to collaborate, reach out to Miller or me, and we can talk about putting some early thing, and then you take it to the audience and connect and refine it. So there are easier platform we have built. So that's one of the advancement we have. We have a product team and we have the service team, but we do a lot of prototype just to check out various aspect of medtech area, healthcare area, to just build something and go out and try out, and then you say, is it there is a product there or there is some technology there? So.
Right.
Thank you so much, Anju. And we are open for one more questions. If anyone has.
Raise your hand, then we can unmute you, or if you want to put that question in the, also we can take it.
Yep. Yes, give me 1 sec. Yes, Gigi, I think you can talk right now. Hope I pronounce it right.
You had to maybe unmute yourself.
Yeah, it's all. Yeah, you have to unmute.
Okay.
Hi. Hello.
Yes.
Hello.
Thank you very much for the conference. I have a question for the professors, for the doctors. Is there any open source code repository that in which we can search for the algorithms that they have been working with? And my other question would be like, are you working with any quantum computing algorithm, or are you developing that? Thank you so much.
Yeah, so many of these algorithms that I talked about are open source. I talked about some of the machine learning models, deep variant, deep consensus. I believe those are open source. And then the other thing, if you wanted to look at some pipelines, I'm happy to send you links if you want to send me email. But if you would look for the GATK reference pipelines in GitHub, GATK, that would be a place to start to look at pipelines. It's a pretty big collection of some standard pipelines and workflows, and there are pointers to reference data sets. But that would be a start. But it's a very large field. So there's many open source algorithms for these different types of analyses, open source pipelines, and open source data sets, so that you can check out and run pipeline or run an algorithm and observe its results. There you go. Yeah.
Oh, thank you so much for sharing
Yeah,
that. Like usually also I work in research field. I'm a pediatric surgeon, and I'm currently doing an artificial intelligence program here in Miami. I'm enrolling in artificial intelligence programs, and we're trying to partner with some of the hospitals here and trying to do some research and collaboration with them. But we're not very sure like we can see a lot of code and algorithms in GitHub, but we don't know which will be like trustable, you know.
Yeah, and I can give 2 other pointers you might look at are the Broad Institute, BROAD, and there's another one that I want to send you a pointer to someone. I can't.
We can follow up.
Email. You can send me email. I'll get that to you.
Yeah, so you can reach out to us, and we will provide some of the thing. I think as you are working on this area, Tom, as we work on, there are other solutions also, like parallel file systems, and there are acceleration capability for technology for universities and research. So we develop and we advise and we provide hardware solutions, software solutions to identify where to go. So if you guys are interested, you can reach out to Tom Miller or me, and we can have a offline conversation and see how it is, because I think Tom has so vast knowledge over last 25-30 years. So this information of pointing out few things is difficult, but you can find out. Okay.
Thank you so much.
Yeah, thank you.
Yeah. So finally, unfortunately it's time. So thank you so much, Dr. Tom. It's been a feast. So I was
happy to be here. I appreciate the opportunity.
Really filled with knowledge, and it's a new endeavor for me. Like, it's a really eye-opener. So, and thank you so much for the active participation for all the audience who join us today. We have covered a lot of ground today, and from the role of AI in genomics and sequencing and availability of technology, and some interesting questions in the end. So it's a really fantastic event. So we can definitely address more, and we can connect people with you in later stages, Dr. Tom. And so after this, we will be reaching out every single one of you who attend today, and the recordings will be shared, and we will be sharing more perks along with it as a courtesy from our end. So thank you so much, wherever you are. Thank you so much, and have a great day.
Thank you for joining.
Bye-bye.
Bye.
Bye.